Hallmarks of cancer. Distinct and complementary capabilities enable tumor growth and metastatic dissemination. These properties of cancer cells have been given as 8 hallmarks of cancer. These hallmarks provide a solid foundation for understanding cancer biology. Figure 4.82. Qualitatively distinct capabilities are contributed by each of these 8 hallmarks, which seem to form an integral part of most types of lethal human cancers. The relative importance and balance of their respective contributions to pathogenesis of disease will certainly vary among cancer types, such that certain hallmarks may be of minor importance or maybe in some cases even absent. Despite these variations with respect to their contributions to disease pathogenesis, there is adequate reason to postulate these as a general phenomenon and hence can be applied towards understanding the biology of human cancer. Figure 4.828 Hallmarks of cancer. These are the functional capabilities that are characteristic of cancers, and is believed that they are acquired by cancer cells through the process of multi-step carcinogenesis that leads to most cancers. The order of acquisition of these hallmarks and the relative balance and significance of their contributions, however, leading to a dramatic increase in p53-mediated transcription. Li-Fraumeni syndrome (LFS) is a disorder caused due to inheritance of a p53 mutation, thus greatly increasing the risk of developing several cancer types: breast cancers, sarcomas, brain tumors, and acute leukemias. Mutations in p53 have been reported in colon, breast, lung, and liver cancers. The frequency of involvement of p53 in neoplasia is higher than other known tumor suppressors or dominant proto-oncogenes. Properties of cancer cells. Over several decades of studies on various cancer types have shown that for cancer cells to sustain as pathological tumors, it is required that they acquire certain unique abilities lacking in normal cells. Normal cells evolve into cancer cells by progressively acquiring these hallmarks in a multi-step process. They attain the tumorigenic and malignant properties, which are typically shared by nearly all cancer types. Studies spanning several decades have enabled the ascertainment of a defined set of characteristic features. These are called the hallmarks of cancer. The following are the established hallmarks of cancer. 1. Ability to sustain proliferative signaling. 2. Ability to evade growth suppressing signals. 3. Ability to resist cell death. 4. Attainment of replicative immortality. 5. Ability to induce angiogenesis. 6. Attainment of invasive and metastatic abilities. In addition to these, 2 more aspects have been considered to be significant, and these are considered as the emerging hallmarks of cancer. They are 1. Ability to reprogram the cellular energy metabolism and 2. Evasion of destruction by the immune system. Underlying all these hallmarks and paving the way for their acquisition are genomic instability and inflammation. Genomic instability facilitates the acquisition of these hallmark traits by creating more genetic diversity in the form by interfering with growth signals. The ability of cells of the immune system to recognize specific tumors was shown in the 1940s by experiments conducted on mutagen-induced murine tumors. It was observed that primarily CD8+ cytotoxic T cells played a primary role in rejection induced by tumors in mice. This further led to the discovery of genes encoding tumor rejection antigens located on murine tumors. Subsequently, antigens recognized tumor-reactive T cells of humans were also identified. Identification of non-mutated tumor antigens, which are broadly shared, led to the prospect of developing effective vaccine therapies against cancers. However, response rates in cancer vaccine clinical trials targeting these antigens have been very low. Successful prevention of cervical cancer establishment has been achieved by vaccination with HPV protein-expressing virus-like particles. Immunization with HPV-6 and E7 protein-derived peptides have been shown to cause regression of high vulvar neoplasia in women. Immune-based therapies leading to regression of large, established metastatic tumors have been identified. Administration of interleukin-2 (IL-2) can cause non-specific immune stimulation, which can further lead to objective clinical responses in melanoma and renal cancer patients. Furthermore, CTLA-4 PD-1-mediated regulatory pathway inhibition can lead to tumor regression in metastatic lung cancer and melanoma patients. Melanoma-reactive T cells, when adoptively transferred to melanoma patients, can mediate objective clinical responses in 50% to 70% of patients. Also, the ability for genetic modification of anti-tumor lymphocytes is making possible the treatment of cancers of other histological origins via cell transfer therapy. Active research aimed at finding potent tumor rejection antigens and mechanisms underlying the regulation of immune responses against cancer are being extensively pursued. Various therapies were initially evaluated in model systems and then developed to treat cancer patients. The therapeutic approaches seems to vary among the different cancer types. Treatment of cancer. The first line of cancer therapy typically includes surgery, radiation therapy, and chemotherapy. In most cases, a combination of these is used to obtain favorable therapeutic outcome. In addition, increasing knowledge with respect to the molecular basis of cancer has led to more effective targeted therapies such as immunotherapy, gene therapy, etc. Radiation therapy. Radiation therapy, also known as radiotherapy, abbreviated as RT, RTX, or XRT, is a therapy that utilizes ionizing radiation for controlling or killing malignant cells. Radiation oncology is a branch of oncology that deals with radiotherapy. Wilhelm Röntgen, in 1895, proved the beneficial use of radiation through his experiments. He discovered the penetrance of X-rays through materials, which were otherwise impenetrable by light. In 1896, the therapeutic use of X-rays was demonstrated by Emil Graup while treating an ulcerated breast cancer in an advanced stage. Since then, great progress has been made, mainly due to research advancements in radiation physics, chemistry, and biology. Cancer immunotherapy. Progress in cancer immunotherapy relied upon the understanding of cellular immunology and tumor-host immune interactions. This led to the development of immune-based therapies that could target human cancers. Studies of transplanted syngeneic tumors and allografts in mice revealed the role of cellular immunity rather than humoral immunity in the rejection of tissues. Research regarding immunotherapy has been directed towards enhancement of antitumoral immune responses mediated by T cells that identify cancer antigens. Use of antibodies that can bind to growth factors on tumor surfaces contributes to tumor regression, not by directly destroying tumor cells, but rather involve nonspecific direct immunization with various immunogens, and passive transfer, adoptive transfer, activated effector cells, table 4.15. 1. Nonspecific stimulation of immune reactions. Stimulate effector cells, e.g., IL-2 in melanoma and renal cancer. B. Inhibition of regulatory factors, e.g., anti-CTLA-4 in melanoma, anti-PD-1 in melanoma and lung cancer. 2. Enhancement of antitumoral reactions by active immunization to cancer vaccines. 3. Transfer of activated antitumoral cells passively, adoptive immunotherapy. Table 4.15 Cancer immunotherapy approaches. Chemotherapy. Chemotherapy utilizes the application of cytotoxic drugs capable of disrupting the cell cycle and thereby causing the death of proliferating cells. The various mechanism of action involved in the process is affected as outlined in Fig 4.83 and Tab 4.16. The chemotherapeutic agents are grouped into several different categories based partly on their mechanism of action and partly on their chemical nature. Although there are examples of successful regimens using single drugs such as methotrexate in the treatment of early choriocarcinoma, placental cancer, it is more common to use a combination of several drugs. It shows that there is an improvement in remission rates with increasing numbers of drugs used in the treatment of Hodgkin's lymphoma patients. Different cancers are treated with different combinations. An acronym is given to each combination. Several combinations are available. For example, CMF contains cyclophosphamide, methotrexate, and 5-FU. Here cyclophosphamide is an alkylating agent, while the methotrexate and 5-FU are antimetabolites. Since cells are capable of developing different types of resistances individually to each of these compounds, using drugs in combination increases the chance for at least one of the drugs will be effective. All cancers do not show a good response to cytotoxic drugs. Choriocarcinomas, germ cell cancers, leukemias, and lymphomas usually respond well, whereas cancers of the pancreas, liver, brain, thyroid, kidney, and melanomas do not. Figure 4.83 Effective chemotherapeutic drugs related to the cell cycle. Chemotherapeutic agents. Cancers treated. Mechanism of action. Process inhibited. Alkylating agents. Cyclophosphamide. Leukemia, breast DNA synthesis. Cisplatin testis, ovary DNA synthesis. Antimetabolites. Methotrexate. Placenta, breast dihydrofolate reductase. 5-fluorouracil. Colorectal, stomach, breast thymidylate synthase, RNA synthesis. Cytosine ribinocyte. Lymphoma, leukemia. DNA polymerase. Natural compounds. Doxorubicin. Leukemia, breast, lung DNA and RNA synthesis. Vincristine testis, lymphoma. Tubulin polymerization. Taxol. Breast, ovary. Tubulin polymerization. Tab 4.16 Chemotherapeutic drugs and their mode of action. Chemotherapy drugs. Alkylating agents. Alkylating agents disrupt DNA synthesis by forming ducts with DNA bases. Two functional groups are present in most alkylating agents. Each of these functional groups can form intrastrand and interstrand cross links within a DNA double helix by reacting with a DNA base. These cross links can be formed at any cell cycle stage, and hence these agents are not specific to any cell cycle phase. 1. Cyclophosphamide. Metabolic activation of cyclophosphamide causes guanine in ducts which mediate its effects. Metabolites of cyclophosphamide block the cell cycle at several stages are combined with 5-FU and methotrexate for the treatment of several cancers, e.g., breast cancer. Side effects of the drug include immunosuppression, sterility, and hair loss. Ifosfamide is a related compound whose effectiveness is equivalent to cyclophosphamide but varies in its toxicity profile. 2. Cisplatin within the body. Cisplatin and carboplatin, its analog, are converted to alkylating species. These preferentially form ducts at the N7 position of adenine and guanine. Further, these ducts interact with adjacent bases within the same strand or another strand, forming intrastrand or interstranded ducts, respectively. The preference order for duct formation is G and GT, A, G and GT, others. DNA helix is distorted with intrastranded ducts, while strand separation is prevented by interstrand links, such that DNA replication is abnormal or altogether inhibited. Cisplatin is effective, particularly against testicular and ovarian cancers, and since it has minimal effect on bone marrow, it is advantageous over other chemotherapeutic agents. Chemotherapy drugs. Antimetabolites. Nucleic acid synthesis is inhibited by antimetabolites, and the mechanism of action is dependent on the compound. 1. Methotrexate. Methotrexate is a folic acid derivative and a competitive inhibitor of the enzyme dihydrofolate reductase (DHFR). DHFR is required for the biosynthesis of purine and pyrimidine and hence for DNA synthesis. The ability of methotrexate alone to cure cancer called choriocarcinoma makes it a unique drug. However, more commonly, it is combined with other therapeutic agents in treatment regimens. Figure 4.84. Inhibition of DNA synthesis by methotrexate via inhibition of dihydrofolate reductase. 2. 5-fluorouracil. 5-fluorouracil, 5-FU, a uracil derivative, can be phosphorylated and incorporated into RNA but is non-functional. In addition, 5-FU produces 5-fluorodump, which inhibits DNA synthesis by forming an inactive ternary complex with thymidylate synthase in the N5, N10-methylene FH4. 5-FU has been mainly used in the treatment of stomach and breast cancers. It is mainly toxic to the bone marrow cells. Figure 4.85. Inhibition of DNA synthesis by 5-fluorouracil via inhibition of thymidylate kinase. Radicals, single and double strand breaks and bind onto topoisomerase 2 are some of the effects. These have been mainly used in the treatment of leukemias and breast cancers. Apoptosis. Cell proliferation is decisive for the growth of multicellular organisms. It all begins from the zygote stage, and development is helped with the rapid proliferation of embryonic cells, which further differentiate to produce a specialized adult tissues and organs. Starting from a single cell, diversification of cell types to produce and organize it into tissues and organs. The process of differentiation of cell involves not only cell proliferation, but also cell death. Programmed cell death, called apoptosis, it is a natural part of development. It plays a vital role in the development and tissue homeostasis. During the development of various cells or in excess, they undergo programmed cell death and sculpturing of organs and tissues. Apoptosis help in the sculpturing of the organism, like generating the middle ear space, the vaginal opening, and the formation of our fingers and toes. The term apoptosis was first used by Kerr, Willie, and Curry in 1972 to define a morphologically noticeable form of cell death. It was first observed in the nematode C. elegans, in which it was observed that exactly 131 cells die according to the normal developmental pattern. The cells of the nematode are programmed to die. Different tissues use different signals for apoptosis. Apoptosis is carefully regulated in the embryonic stage to ensure the development of the organism. In adults, it is crucial for balanced growth. It provides a defense mechanism by eliminating of damaged and potentially dangerous cells. Most of the virus-infected cells undergo programmed cell death. Other factors initiating programmed cell death are traumatic events, like exposure to toxic chemicals, biotic and abiotic stressors. DNA damage checks the potentially harmful mutations to propagate further, which might lead to the development of cancer. Abnormalities of cell death may. 3. Cytarabine, Ara-C. Ara-C is formed when arabinose, another 5-carbon sugar, is introduced in place of deoxyribose in cytidine. Phosphorylation of Ara-C forms Ara-CTP, which inhibits DNA polymerase alpha. Some Ara-C also gets incorporated into DNA. Blockage of DNA synthesis occurs by both these processes, and hence Ara-C functions as an S-phase inhibitor. It has been effectively used against leukemia, and it is mainly toxic to the bone marrow. Chemotherapy drugs. Natural products. 1. Topoisomerase inhibitors. Topoisomerase enzymes are involved in the uncoiling of supercoiled DNA during and after replication. There are two major types of topoisomerase I and II. Inhibitors are available for both types of topoisomerases. Topoisomerase I is inhibited by camptothecins, such as topotecan, and topoisomerase II is inhibited by apophatophyllotoxins, such as adipocyte. Double-strand breaks are caused by both inhibitor types such that it results in cell death, i.e., they induce G2 arrest and apoptosis. 2. Microtubule poisons. Disruption of spindle function during mitosis is caused by these drugs. Due to this, separation of chromosomes does not take place. Cell division is blocked at metaphase, followed by apoptosis. Examples include vinca-alkaloids, vincristine, vinblastine, vinorelbine that prevent tubulin polymerization, taxanes, paclitaxel, docetaxel that prevent depolymerization by stabilizing the microtubule. In both situations, abnormal spindles are formed, and apoptosis occurs. A wide spectrum of cancers, leukemias, lymphomas, ovarian, breast and testicular cancers are responsive to these compounds. 3. Anthracycline antibiotics. Antibiotics such as doxorubicin and epiroubicin have numerous effects on DNA. The partial unwinding of the double helix due to intercalation, generation of free and term release of cytoplasmic contents into the surroundings, initiating an inflammatory response. As the apoptotic cells do not spill their cellular content and are readily phagocytosed, hence there is no inflammatory response. The event of programmed cell death is a highly controlled systematic process which is regulated by a group of the gene. These genes and their molecular mechanisms underlying in apoptotic pathways are found to be evolutionarily conserved. Onset of apoptosis was believed to be an irreversible process until recent times. However, the initiation and thereafter clearance of apoptotic cells by macrophages involve various molecular factors. It has been experimentally illustrated in C. elegans embryos that blocking engulfment genes enhances cell survival in the case of weak pro-apoptotic signals. In C. elegans, mutations of killer genes causing partial loss of function to allow the survival of some cells that faded to apoptosis, whereas mutations in engulfment genes in turn enhance the frequency of this cell survival. However, mutations in engulfment genes alone allow the survival and differentiation of some cells that were otherwise destined to die via apoptosis. These findings suggest that genes that conciliate corpse removal also facilitate the active killing of the cells. In the vertebrate model, there is a study about the potential role for macrophages in enhancing the death of cells in some tissues. Eradication of macrophages from the anterior chamber of the radi leads to the survival of vascular endothelial cells, which normally undergo the events of apoptosis. The process of apoptosis is highly complex and tightly regulated. It involves an energy-dependent cascade of molecular events. For the occurrence of apoptosis, there are two major pathways, the extrinsic pathway and the intrinsic pathway. However, both pathways are linked and influence each other. Other than these, there is an additional pathway, which involves T-cell-mediated cytotoxicity and perform granzyme-dependent mechanism. The extrinsic, intrinsic, and granzyme B pathways. Because of a variety of illnesses, such as cancer, autoimmune disease, and neurodegenerative disorders, the detailed mechanisms of cell death regulation are growing areas of research in medicine. Programmed cell death. Programmed cell death plays a vital role in the regulation of associations between cells of tissues. There is an alternative to apoptotic cell death, i.e. necrosis. It is considered a toxic process. It is an energy-independent process of cell death. Apoptosis is clearly different from necrosis. In necrosis, the cells suffer major damage, leading to a loss of membrane integrity and disruption of the cells. During necrosis, the cellular contents are released uncontrolled into the cell's environment, which results in damage to surrounding cells and a strong inflammatory response in the corresponding tissue. Necrosis has characteristic stages like oncosis, karyolysis, and cell swelling. However, in apoptosis, it exhibits cell shrinkage, pycnosis, and karyorrhexis. Necrosis is accompanied by cell swelling. Though their mechanisms and morphologies differ, there is an overlap between apoptosis and necrosis processes. There is evidence indicating that necrosis and apoptosis are interconverted. There are factors that convert a progressing apoptotic process to a necrotic process. It includes a decrease in the amount of caspases and intracellular ATP. Necrosis or apoptosis pathway is dependent on the signal, the tissue type as well as the developmental stage of the tissue. Necrosis is an uncontrolled which affects a large number of cells. On the contrary, apoptosis is a controlled process which affects only an individual or clusters of cells. Necrotic is mediated by some important mechanisms: hindered energy supply of the cell and damaged cell membranes, cell swelling, also formation of cytoplasmic vacuoles, even distended endoplasmic reticulum as well as cytoplasmic webs along with condensed, ruptured mitochondria, disaggregation and removal of ribosomes and ruptured lysosomes, leads to disruption of the cell membrane which converge to the same execution pathway. They are initiated by the cleavage of an active proteolytic coenzyme PROCASPASE3 to caspase 3, causing DNA fragmentation, degradation of cytoplasmic and nuclear proteins, formation of apoptotic bodies, and stimulating phagocytosis. Different morphological and biochemical changes occur in the apoptotic cell. Figure 4.86. The apoptotic pathway in mammalian cells are more complex than C. elegans. Mechanism of apoptosis. In 1986, Robert Horvitz and his team discovered that C. elegans carrying a mutation in the gene CED3 failed to perform apoptosis during development. This indicated the product of the gene CED3 is important for apoptosis in this organism. Later on, researchers found homologous genes in other organisms. After cloning and sequencing of the CED3 gene, it was found that it encoded a protease, providing an insight into the molecular process of apoptosis. Now it is well known that CED3 is a prototype of a caspase family of proteases, known as caspase because they have a cysteine C residue present at the active sites of enzyme, and it cleaves after aspartic acid residues of the substrate proteins. Various caspases have different recognition sites which involve recognition of a different set of neighboring amino acids. Caspases are the eventual executioners of the apoptosis. Caspase has nearly 100 different target proteins in the cell. Caspases mostly target the inhibitors of the DNAs, which in turn are activated and cleave nuclear DNA. They also target cytoskeletal proteins, leading to disruption of the cytoskeleton and causing cell membrane cleaving. As in C. elegans, only one caspase, CED3, is present. In Drosophila and mammals, 7 different caspases are isolated, having a role in various aspects of apoptosis. These caspases are categorized as either initiator or effector caspases. They together bring about cell death. Caspases are synthesized as inactive precursors, which are activated by proteolytic cleavage, catalyzed by other caspases. The activation of an initiator caspase starts a chain reaction leading to a cascade of downstream effector caspase activation, and ultimately death of the cell. Therefore, regulation of caspase activation is vital for determining cell survival. In higher organisms, activation and regulation of apoptosis is more complex and depend on corresponding analogous components found in C. elegans. In mammals, an array of external signals can trigger any of the two major apoptotic pathway, the intrinsic pathway, the mitochondrial pathway, or the extrinsic pathway, death receptor pathway within a cell. The extrinsic pathway is triggered by stimuli such as the binding of death-inducing ligands to cell surface receptors. Whereas apoptosis is also initiated by intrinsic signals, including DNA damage induced by irradiation, chemicals, growth factor deprivation, or oxidative stress. Generally, intrinsic signals initiate apoptosis via the involvement of the mitochondria. Initially, CED4 was presumed to be an activator of the caspase CED3 in C. elegans. Later on, studies illustrated that CED4 is homologous to mammalian APAF1. It binds to caspase C3 and promotes their activation. C. elegans as a model system contains the basic components of the cell death machinery. Whereas in mammalian cells, caspase CASPASE9 is a key initiator, activated by binding to APAF1, forming a multi-subunit complex known as the apoptosome. Figure 4.87. The activation of BCL family protein in apoptotic cells. In mammalian cell, apoptosome requires cytochrome C, released from mitochondria by apoptotic signal. Once apoptosome is activated, it initiates the cascade by activating CASPASE9, which activates other downstream effector caspases, resulting in cell death. There are about 14 mammalian caspases have been identified. All caspases have a number of common features. Zymogen's procaspase containing a protamine followed by P20 large and P10 small subunits. These zymogens can be cleaved to form active enzymes following the induction of apoptosis. All caspases can cleave substrates at the asp XX bond, which is unique among mammalian proteases, except for the serine protease granzyme B. They are categorized into groups like initiators CASPASE2, -8, -9, -10, effectors or executioners CASPASE3, -6, -7, and some as inflammatory caspases CASPASE1, -4, -5. There are other caspases also identified. It includes CASPASE11, which has a role in apoptosis regulation and maturation of cytokine during septic shock, CASPASE12, which mediates the endoplasmic-specific apoptosis and the cytotoxicity induced by amyloid beta. Caspase 13 is a gene expressed in bovines. CASPASE14 is specifically expressed in embryonic tissues and not in adult tissues. Extensive protein cross-linking is a characteristic feature of apoptotic cells, which is achieved through the expression and activation of tissue transglutaminase. CA2+ and MG2+ dependent endonucleases also cleave the DNA fragments of 180 to 200 base pairs. It can be visualized as a DNA ladder on agarose gel electrophoresis. Caspases accomplish apoptosis by activating various hydrolytic enzymes including the group of essential proteins. Some of the major targets of caspases are as follows: I. Various protein kinases, such as focal adhesion kinase FAK, PKB, PKC, and RAF1. Disruption of FAK inhibits the cell adhesion and detachment of the cell from its neighbors. Lamins form the inner lining of the nuclear envelope. Disintegration of lamins leads to the disfiguration and shrinkage of the nucleus. Proteins forming the cytoskeleton, especially those of intermediate filaments, actin and tubulin, are inactivated, leading to changes in cell shape. An endonuclease called caspase-activated DNase (CAD) is activated after cleavage of its inhibitory protein. Activated, CAD is translocates to the nucleus from the cytoplasm, where it attacks DNA. Apoptosis can be set off by various internal stimuli and external stimuli like abnormalities in the DNA and certain cytokines. For example, prostate epithelial cells become apoptotic in absence of testosterone. The external stimuli that activates the apoptotic process are called the extrinsic pathway, whereas initiation due to internal stimuli is called the intrinsic pathway. Both the extrinsic and intrinsic pathways are discussed separately later in this chapter. However, there is crosstalk between intrinsic and extrinsic pathways, and extracellular apoptotic signals may cause activation of the intrinsic pathway also. Figure 4.88. The structure of TNFR, tumor necrosis factor receptor. The TNF molecule is attached to the TNF receptor on the extracellular region. The attachment of the ligand to its receptor brings about a conformational change in the intracellular domain of the TNF. It activates the death domain, which in turn proteolytically cleaves the procaspase 8 into active initiator caspase 8. Extrinsic pathway. Dr. P. Bruns, a German physician, gave the idea of the extrinsic pathway's existence leading to apoptosis in 1868. He reported that under acute bacterial infection, tumor retrogression is observed in some patients. Nearly a century later, lymphotoxin and tumor necrosis factor (TNF) were isolated, and the role in the effective killing of cultured tumor cells was established. The apoptosis induced by the external factor is called the extrinsic pathway. It can be induced through various factors. The extrinsic pathway is illustrated in stepwise order. Fig. 4.89. The generalized sequence of events is similar to most of the signaling pathways. After the binding of the ligand, the receptors undergo conformational changes facilitating the recruitment of an assembly of proteins, also known as the death-inducing signaling complex disc. It was first described in facile phase apoptotic signaling. In the case, the stimulus for apoptosis is tumor necrosis factor (TNF), an extracellular messenger protein named after its ability to kill tumor cells. In certain cells of the immune system, TNF is produced in response to hostile conditions, such as exposure to ionizing radiation, viral infection, elevated temperature, or toxic chemical agents. Phos plays a vital role in the regulation of apoptosis of immune cells. Phos-facile interactions regulate the immune system in various ways. Phos is involved in T cell-mediated killing, inflammatory and immune cells destruction in immune-privileged sites, and elimination of self-reacting B cells and activated T cells left over after an immune response. Levels of serum FASL have been seen elevated in patients' diseases such as NK cell large granular lymphocyte leukemia, systemic lupus erythematosus, rheumatoid arthritis, Sjogren's syndrome, lymphohistiocytosis, myocarditis, and acute graft versus host disease. Some tumors have been reported to express FASL, a mechanism possibly to evade attacking lymphocytes. FAS were mapped to the chromosomal location of a lymphoproliferative disorder known as LPR in the mouse. A point mutation in FASL near the extracellular carboxyl domain gives rise to the GLD phenotype. Both LPR and GLD mice fail to eliminate excess lymphocytes and display a lymphoproliferative phenotype such as lymphadenopathy and splenomegaly. Figure 4.89 The initiation of intrinsic factor is majorly dependent on the release of cytochrome C, while the extrinsic pathway is dependent on death receptor DCR-FOS and TNFR. TNF binding to a transmembrane receptor, tumor necrosis factor receptor TNFR1 initiates its response. The TNF receptor is present as a trimer in the plasma membrane. The cytoplasmic domain of the TNF receptor subunit has a 70 amino acids peptide chain, also called as death domain, that facilitates the protein-protein interactions. Binding of TNF to the receptor causes a change in the conformation of the receptor's death domain, which facilitates the recruitment of various other proteins. DR4, DR5, and FOS go through conformational changes leading to the assembly of the death-inducing signaling complex disc. Ligand binding to the decoy receptor DCR1, DCR2, and DCR3 have high affinity, but it does not induce apoptosis. DR4, DR5, and FOS recruits the FOS-associated death domain FADD through complementary death domains DDs. FADD can recruit caspase 8 with their complementary death effector domains DEDs. This leads to its autoproteolytic cleavage of caspase 8, releasing two subunits that form the active enzymes. In the inner surface of the plasma membrane, at last, two PROCASPASE8 molecules join to complete the assembly of the protein complex. These proteins, procaspases, is a precursor of a caspase. They contain an extra segment of peptide which must be cleaved by the proteolytic process to activate the enzyme. The synthesis of proenzymes of caspases helps in keeping check from accidental damage and proteolytic damage to the cell. Even as proenzymes, the procaspases exhibit a low level of proteolytic activity. When two or more procaspases are in close association with one another, they initiate proteolytic cleaving of one another, leading to the fully active caspase. The processed active enzyme, i.e., CASPASE8, contains 4 polypeptide chains derived from 2 procaspase precursors. Activation of CASPASE8 is similar in principle to the activation of effectors by a hormone or growth factor. Like in each signaling pathway, the extracellular ligand receptor interaction causes a change in the conformation of the receptor molecule, facilitating the binding and activation of other client proteins in the downstream. Intrinsic pathway. An irreparable genetic damage, extremely high concentrations of cytosolic CA2+, viral infection, or severe oxidative stress trigger apoptosis by the intrinsic pathway. It is not triggered by receptor-mediated pathway. It is an intracellular pathway which depends upon various positive as well as negative signals. This, in turn, leads to enhanced membrane permeability. Thus, membrane permeability transition (MPT) pores are open and augment the pro-apoptotic proteins into cytosol. Some of these proteins include cytochrome C, SMAC Diablo, and HTRA2 OMI. Cytochrome C, which binds with APAF1 and procaspase 9, is one of the most important factors responsible for triggering the intrinsic pathway. The complex of cytochrome C, APAF1 and procaspase 9 is called apoptosome. Cytochrome C is the main component required for CASPASE9 activation. The activation of the intrinsic pathway is regulated by proteins of the BCL2 family of proteins. Member of the BCL2 family have characteristic features, i.e. presence of one or more BH domains. Figure 4.90 Mitochondrial regulation of apoptotic cell death. The permeability transition for PT is the BDAC and the adenine nucleoside translocator and main components of the intrinsic pathway initiation through mitochondria, which are adjacent in the OMM and intermitochondrial membrane IMM respectively. Along with these proteins, in the OMM, peripheral benzodiazepine receptor PBR and BAX-BCL2 and in the IMM, cyclophilin D and cardiolipin complete the PT complex. During normal conditions, the PT works as a sensor for the mitochondrial matrix. It senses the matrix pH, matrix CA2+, gate potential, redox state, and others. The BDAC is permeable for the solutes of up to 5 kDa, and along with the help of mitochondrial creatine kinase (MTCK), it shuttles substrates such as ATP from the mitochondria. The ANT, on the other hand, is impermeable, which helps to generate the electrochemical potential for oxidative phosphorylation. The big apoptogenic proteins such as cytochrome C 12 kDa, a mitochondria-derived activator of caspases direct inhibitor of apoptosis IAP, binding protein with low pi, SMAC Diablo, 100 kDa AIF, 57 kDa, and others are predominantly stored in vesicles created by infoldings of the IMM intercrystal space. During the onset of apoptosis, the ANT conductance and permeability increase leading to osmotic swelling of the mitochondrial matrix, releasing the apoptogenic proteins from their intercrystal storage into the intermembrane space. The interaction of BDAC with BCL2 protein leads to enhanced permeability and release of cytochrome C. The release of cytochrome C from mitochondria initiates the intrinsic pathway. Various pro-apoptotic and anti-apoptotic proteins regulates the programmed cell death. They can be subdivided into 3 groups. 1. Pro-apoptotic members, which promote apoptosis, e.g., BAX and BAC. 2. Anti-apoptotic members that check the apoptosis, e.g., BCLXL, BCLW, and BCL2. 3. BH3 only proteins. They share a single small domain of the BH3 domain with other BCL2 family members. It promote apoptosis by an indirect mechanism. Fate of any cell is determined by the BH3 proteins and whether a cell is going to the pathway of survival or death. BH3 proteins, e.g., BID, BAD, PUMA, and BIMP show their pro-apoptotic effect through 2 different ways. Either they promote apoptosis by anti-apoptotic BCL2 members inhibition, or promote apoptosis by pro-apoptotic BCL2 members activation. It is vital for the health of a cell that BH3 proteins are either strongly inhibited or absent. The anti-apoptotic BCL2 proteins checks the activation of pro-apoptotic members. The mechanism is still not clear. It is observed during certain types of stress conditions that the BH3 proteins start getting expressed or activated and taking a cell towards the direction of apoptosis. Under such situation, the governing effects of the anti-apoptotic BCL2 proteins are quashed, and the pro-apoptotic factors of the BCL2 family, such as BAX, gets translocated to the outer mitochondrial membrane from the cytoplasm. Entire mechanism of translocation is not very clear. One proposed hypothesis suggests that BAX molecules undergo conformational change facilitating their insertion into the outer mitochondrial membrane, which in turn initiates their assembly into a multi-subunit channel. Formation of this channel drastically enhances the permeability and movement of molecules across the outer mitochondrial membrane, such as cytochrome circa. Increased mitochondrial membrane permeability can occur with an increase in cytosolic CA2+ levels released from the ER. Figure 4.91 Various proteins of BCL2 superfamily and their pro and anti-apoptotic activity. Cytochrome C is an intermembrane protein. Enhanced permeability can release all the cytochrome C molecules present within 5 minutes of every mitochondrion of the cell. Release of cytochrome C in the cytoplasm is considered as the point of no return that irreversibly leads the cell to apoptosis. The cytochrome C released in cytoplasm forms a multiprotein complex known as the apoptosome. It is a complex of cytochrome C, procaspase 9, and various other molecules. Apoptosome activates the PROCASPASE9 into caspase 9 molecules without proteolytic cleavage, unlike CASPASE8. Caspase 9 is an initiator caspase that activates other downstream executioner caspases, facilitating apoptosis. The extrinsic and intrinsic pathways post-initiation gets ultimately converged. Down the line, the same executioner caspases activation leads to cleavage of the same cellular targets. As cells undergo apoptosis, they lose contact with their adjoining cells and start to shrink. Ultimately, the cellular component disintegrates, and the cell condenses to form the membrane-enclosed apoptotic body. This entire process can be executed in less than an hour. The apoptotic bodies attract and are recognized by phagocytic cells due to the presence of phosphatidylserine on their surface. It is a phospholipid normally present only on the inner leaflet of the plasma membrane. In the course of apoptosis, an enzyme, scramblase, moves phosphatidylserine molecules from the inner leaflet to the outer leaflet of the plasma membrane. Phagocytic cells recognize them as eat me signals. Apoptotic cell death thus is clean death, i.e. cellular content is not spilled out into the extracellular environment. Spillage of cellular debris may trigger an inflammatory reaction, which can cause additional tissue damage. Of the execution phase of apoptosis, both the mitochondrial pathway and CASPASE3 activation are crucial for granzyme B-induced killing. Studies suggest that this method of granzyme B cytotoxicity has an important role in the regulation of T-cell expansion of type 2 helper Th2 cells. Experimental studies establish that death receptors and caspases are not involved with the T-cell receptor-induced apoptosis of activated Th2 cells, as even after blocking their ligands, apoptosis was not affected. Whereas phos-phos ligand interaction, caspases and death domain adapter proteins are involved in the regulation of apoptosis of cytotoxic type 1 helper cells in which granzyme B has no effect. Granzyme plays an important role in cytotoxic T-cell induced apoptosis. It activates the caspase-independent pathways. Granzyme activates DNA digestion with the help of a tumor suppressor gene product DNase NM23H1. This DNase NM23H1 plays an important role in the weeding of cancer through the induction of tumor cell apoptosis by immune cells. The NM23H1 gene is inhibited by a 270 to 420 kDa endoplasmic reticulum-associated complex, SEC complex containing the nucleosome assembly protein set. Granzyme cleaves the SEC complex, resulting in DNA degradation leading to apoptosis. The SEC complex has a role in chromatin structure and DNA repair. This complex is made up of SET, ATE1, PP32, and HMG2. It seems to work together to protect chromatin and DNA structure. Hence, the lysis of this complex by granzyme disrupts the maintenance of DNA. Perforin pathway. In type 4 hypersensitivity, TC cell with sensitized CD8+ cells kill cells bearing the antigen. Natural killer NK cells and cytotoxic T lymphocytes (CTLs) accomplish to kill target cells through various methods, i.e. the extrinsic pathway, or phos-phos ligand interaction pathway, predominantly to induce apoptosis. Other than those, they also cause apoptosis of tumor cells as well as virus-infected cells through the perforin pathway. There is synergy between perforin and granzyme on the basis of ultrastructural analyses. When purified perforin was added to inert membrane preparations, e.g. lysored erythrocytes, the initial observation concurred with that polyperforin pores induce osmotic instability and target cell lysis. The antigenic similarity between perforin and complement component 9 was reinforced. The perforin molecule is secreted from NK cell (CTLs), which is transmembrane pore-forming protein. Pore extends into the target cell and releases the cytoplasmic granules through the porin into the target cell. The granules are serine proteases called granzymes. The most important component within the granules are granzyme A and granzyme B. Granzyme B cleaves the proteins at aspartate residues, and therefore, it activates PROCASPASE10 and may cleave factors like ICAD, inhibitor of caspase-activated DNase. Various studies have also shown the role of granzyme B in the mitochondrial pathway. In turn, it amplifies the death signal by the release of cytochrome C and specific cleavage of BID. It also may activate CASPASE3. It leads to the direct induction. Figure 4.92 different apoptotic pathways. Major apoptotic pathways are intrinsic and extrinsic as well as granzyme pathway. Each pathway differ in trigger signal, and their initiator caspase 8, 9, 10 while all pathway progresses through energy-dependent common execution pathways to caspase 3. Physiology of apoptosis. Apoptosis is important for the elimination of damaged cells. Cells with altered genomes are particularly dangerous because of the chances that mutations that occurred may lead to the development of cancer. One of the principal triggers of apoptosis is DNA damage, helping in the elimination of potentially harmful cells. Several checkpoints of the cell cycle halt the cell cycle. Progression if any damaged DNA is found. This allows time for the repair. In mammalian cells, in response to DNA damage, transcription factor P53 arrests the cell cycle. The damaged DNA activate ATM and CHK protein kinases. It phosphorylates and stabilizes P53 in turn. The results into increased half-life of P53 leading to transcriptional activation of P53 target genes. The P53 target genes include the CDK inhibitor P21, which inhibits CDKL, cyclin and CDK2, cyclin complexes and stops the cell cycle. The activation of P53 by DNA damage can also induce apoptosis. The P53 also activate the transcription of genes encoding pro-apoptotic BCL2 family members, like PUMA and NOXA. Hence P53 mediates can cause cell cycle arrest and apoptosis as a response to DNA damage. The extent of DNA damage determines the level of P53 and the fate of a cell. Whether the cell leads to apoptosis or reversible cell cycle arrest, it is also dependent on the survival or death signals being received by the cell. As discussed already, apoptosis is vital shaping of an organism, physically and physiologically. For example, role of programmed cell death in the development of the nervous system in the vertebrate. Approximately 50% of neurons die due to a lack of survival signal by apoptosis, while the survivors have received sufficient amounts of survival signals from their target cells. These growth factors related to nerve growth factor (NGF), active survival signals are polypeptide, which stimulates both neuronal survival and differentiation. In contrast, now it is well established that cancer cells evade the normal process of programmed cell death, and their ability to escape apoptosis is more significant than uncontrolled proliferation for their development as malignant tumors. In mammals, a family of approximately 20 genes encodes proteins related to BCL2. They are categorized into 3 functional groups, Fig 4.93. Some of the BCL2 family members, like BCL2 itself, act as inhibitors of programmed cell death and apoptosis. Some members are pro-apoptotic proteins that have role in caspase activation and promote apoptosis. There are 2 major groups of pro-apoptotic proteins, which varies in function and their homology to BCL2. BCL2 and other anti-apoptotic family protein members share 4 conserved regions called BCL2 homology BH domains. One of the group of pro-apoptotic family members called the multi-domain pro-apoptotic proteins consists of 3 BH domains, BHL, BH2, and BH3, whereas another group, the BH3 only proteins, have only the BH3 domain. To the survival signaling pathways that promote cell survival, there are signals to promote programmed cell death. These cell death signals belong to the tumor necrosis factor TNF family. They bind to the TNF receptor family, which stimulates apoptosis in a variety of cell types. One of the best characterized members of this family is the FOS ligand, FOS receptor interaction, which is crucial for the regulation of cell death in the immune system. FOS is responsible for the killing of target cells, such as cancer cells or virus-infected cells, and the elimination of surplus lymphocytes left over after an immune response. Regulators of apoptosis. In mammalian cells, gene called BCL2, an oncogene, was studied. It contributed to the unfolding of human B-cell lymphomas. Unlike other oncogenes proteins, which stimulate cell proliferation, such as ROS, BCL2 is found to inhibit apoptosis. BCL2, as a regulator of apoptosis, focused attention on the consequence of cell survival in cancer development. Figure 4.93 The initiation of apoptosis can occur through either intrinsic or extrinsic pathways. Various pro-apoptotic and anti-apoptotic molecules together regulate the process. Once the initiator caspase is activated, in turn, it activates the executioner caspase, caspase 3, 6, 7. Once the executioner caspase has activated, the fate of the cell is sealed for apoptosis. The fate of the cell that is life or death is dependent on the balance between the activity of pro-apoptotic and anti-apoptotic protein of BCL2 family members, which regulates one another. The multi-domain pro-apoptotic family members, like BAX and BAC, are the downstream effectors which directly induce apoptosis. Their activity is checked with the anti-apoptotic family member interactions, like BCL2. The BH3 only family member proteins are upstream members of the apoptotic cascade. They are regulated by the cell death or cell survival factors. Post-activation, the BH3 only family members repress anti-apoptotic BCL2 family members and activate the multi-domain pro-apoptotic proteins initiating the downstream caspase activation and cell death. In mammalian cells, members of the BCL2 family act at mitochondria, which play a central role in controlling programmed cell death. After activation, BAX and BAC form oligomers assembly in the mitochondrial outer membrane. BAX or BAC oligomers cause the release of cytochrome C into the cytoplasm from the intermembrane space of mitochondrial by forming pores. The cytochrome C from mitochondria then activates the key initiator caspase in mammalian cells, CASPASE9, which forms a complex with APAF1 and forms an apoptosome or apoptotic body. In healthy cells, APAF1 and CASPASE9 are present within the cytosol in an inactive form. The activation of BAX or BAC triggers an apoptosis cascade and leads to the formation of apoptosomes. Caspase activation is also regulated by the IAP, inhibitor of apoptosis protein family. Proteins of the IAP family directly inhibit caspase activity or target caspases for ubiquitination and proteasomal degradation and suppress apoptosis. IAPs are present in both Drosophila and mammals, but not C. elegans, and regulation of their activity or expression provides another mechanism for regulating apoptosis. In mammalian cells, there are other proteins that promote apoptosis. These include 2 proteins called SMAC, Diablo, and OMI HTR2 that interfere with the IAPs. Assays for apoptosis. Since apoptosis occurs due to the interplay of various signaling cascades being tightly regulated through various pathways, these different molecules involved in regulation can be used as a marker for apoptotic assay. However, various apoptosis and necrosis features can be overlapping, hence specific molecules having a crucial role to apoptosis exclusively can be targeted for assays. Assay can be designed to detect the initiation of apoptotic events as well as to detect the later execution event. There is a variety of assays established, each having advantages and disadvantages of its own. Therefore, which one to be chosen depends upon the sample type and objective of the study. Apoptosis in cultured cells line will eventually lead to secondary necrosis. Apoptosis is a quick to process, and cells disappear quickly. From initiation to completion of apoptosis, it takes around 2 to 3 hours. Therefore, the high chance of false negative result is there in the assay if done too early or too late. Apoptosis can occur at localized specific sites within organs, tissues, and cultures. The choice of assay endpoint is critical. It depends upon the mechanism of cell death, the toxin exposure time, and concentration. There are numerous methodologies and assays for apoptosis detection. Some of the most common assays are discussed. Apoptosis assays can be classified into 6 major groups. 1. DNA fragmentation. 2. Detection of caspases, cleaved substrates, regulators, and inhibitors. 3. Membrane alterations. 4. Detection of apoptosis in whole mouse. 5. Mitochondrial assays. DNA fragmentation. In this assay, DNA is extracted from a lysed cell homogenate and further subjected to agarose gel electrophoresis. If the result has the characteristic DNA ladder, with each band in the ladder has a difference of 180 base pairs approximately. The DNA laddering is product of apoptosis caused by endonuclease cleavage in the process. This technique is useful for tissues and cell cultures having high numbers of apoptotic cells within the tissue mass. Besides that, it has a disadvantage with low numbers of apoptotic cells. There are chances this assay may give higher false positive results as the DNA fragmentation occurs in the later phase of apoptosis or necrosis. Hence, this assay should be paired with some more tests to validate the results. Detection of caspases, cleaved substrates, regulators, and inhibitors. There are about 13 characterized caspases potentially used for apoptosis detection. The caspases are used as the marker. The immunohistochemistry assay can detect cleaved substrates such as carbon known cell modifications such as phosphorylated histones. Fluorescent labeled caspase inhibitors can be used to localize the active caspases within cells. Active caspase can be detected in various ways like Western blot, immunoprecipitation, and immunohistochemistry. The major disadvantage with Western blot and immunoprecipitation is the assay does not localize the apoptotic cells or which type of cell is undergoing apoptosis. Just activation of caspase does not always mean apoptosis will occur. Microarray assay of apoptosis is a recent approach that uses the expression profile of at least 112 genes involved in apoptosis. These determine the expression profile of genes that translate into key ligands, receptors, intracellular modulators, and transcription factors involved in the regulation of programmed cell death. Similarly, genes involved in anti-apoptosis are also assayed. Comparison of gene expression in the control and test sample is performed. Hierarchical cluster analysis of genes for temporal expression patterns of transcriptional activation and/or repression is performed. Membrane alterations. Another biochemical marker of apoptosis is the expression of cell surface markers facilitating early phagocytic recognition of apoptotic cells and differentiation from adjacent cells, permitting quick phagocytosis and minimal affect to the surrounding tissue. The movement of the normal inward acing phosphatidylserine from the inner layer to the outer layers of the plasma membrane. Externalization of phosphatidylserine residues acts as eat me, signaling invitation for phagocytic cells. The transfer of phosphatidylserine to the outer side of the cell membrane allows the unidirectional transport of certain dyes. As the cell shrinks in volume, with accumulated dye, the cell can be visualized with light microscopy. Detection of apoptosis in whole mounts. In the whole mounts of embryos or tissues, apoptosis can be visualized using dyes such as acridine orange, AO, Nile blue sulfate, NBS, and neutral red, NR. These dyes are acidophilic. They are stains in the cytoplasmic area of high lysosomal and phagocytotic activity. The disadvantage with this assay is that these dyes cannot distinguish between lysosomes degrading apoptotic debris from the degradation of other debris such as microorganisms. This assay should be cross-validated with another assay. This dye is stable, penetrates thick tissues, and also resistant to quenching. It can be used for a variety of samples such as cell culture, a whole mounts of embryos, tissues, or organs. Mitochondrial assays. In mitochondrial assays, the release of cytochrome C allows the detection of the intrinsic pathway of apoptosis. Techniques like laser scanning confocal microscopy (LSCM) can create submicron thin optical slices through living cells, and several mitochondrial events can be observed in intact single cells over time. The disadvantage with this technique is that the mitochondrial parameters can also occur during necrosis. Cytochrome C can also be assayed using fluorescence and electron microscopy. Limitation of cytochrome C is stability. It is an unstable molecule after it is released into the cytoplasm. Therefore, a control should be used parallel. Other apoptotic or anti-apoptotic regulator proteins can also be detected using fluorescence and confocal microscopy. Basics of the immune system. The human body, like all other multicellular organisms, is constantly under attack from a plethora of pathogens. These pathogens can range from being as small as the poliovirus, approximately 30 nanometers, to being as large as the parasitic kidney worm Dioptifine renal, which can grow to 100 centimeters in length and 10 millimeters in width. To counter this diverse array of invaders, multicellular organisms harbor a highly evolved, extremely versatile and fascinatingly complicated immune system which is indeed capable of providing immunity to most if not all pathogens. The fundamental characteristic of the vertebrate immune system is its astonishing adaptability that allows it to employ a range of mechanisms for the recognition and destruction of a highly heterogeneous multitude of pathogenic invaders. Although certain features of the immune system observed in vertebrates can be seen in various plants and animals, this book will be focused solely on the highly evolved mammalian immune system. The incredible adaptability and effectiveness of the mammalian immune system is no mean feat. To achieve this, it employs an extremely complicated network of cells, organs, molecules, and pathways. Highly dynamic by nature, the immunological infrastructure of the mammalian immune system can be incredibly interconnected and sometimes even circular and confusing in nature. The inherently complex nature of the immune system demands great attention to detail when studying it, with special emphasis on the molecular and cellular players. However, although such studies form the core of understanding and furthering the discipline of immunology, it often masks the larger role immunological research plays in the advancement of human society. Indeed, progress in immunological perceptions can be directly related to landmark events in the history of human civilization, such as the eradication of smallpox through organized and effective vaccination, systematic and risk-free blood transfusions, and transplantation of organs in humans. Such manifestations of basic immunological research continue to motivate and drive further understanding of the mammalian immune system even in the present era. It must be understood that the immune system does not operate in isolation. As we will see in subsequent chapters, the mammalian immune system is intricately linked to the endocrine, nervous, and metabolic systems. Further understanding of the immune system will possibly reveal greater connectivity with the rest of the body and its larger role in the maintenance of homeostasis. Information gleaned from immunological research in the present time will no doubt add great value to the burgeoning field of biomedical science as well as basic science. Humoral and cellular components of immunity. Although Louis Pasteur demonstrated the efficacy of vaccination as a measure to induce protective immunity against a variety of diseases, he could not explain how it worked. Some scientists believe that the immunity induced in vaccinated individuals was bestowed by cells, while others hypothesized that soluble agents were responsible for the same. Experiments carried out by Emil von Behring and Shibasaburo Kitasato in 1890 provided the first insights into the inner workings of immunity. While working together in Berlin, the two scientists reported an astonishing observation. They immunized rabbits with tetanus and subsequently collected the cell-free fluid, i.e. the immune serum from these animals. They had then injected a small portion of this serum into the abdominal cavities of 6 mice and, after They had then injected a small portion of this serum into the abdominal cavities of 6 mice and, after 24 hours, infected them and untreated control mice with live, virulent tetanus bacteria. They observed that extraordinarily all the control mice died within 48 hours of exposure, while not only did the treated mice survive, they did not exhibit any sign of infection. This seminal experiment demonstrated 2 important points: protective agents that grant an organism immunity to a certain pathogen appear in the serum following immunization, and that immunity can be passively acquired, i.e. transferred from one animal to another by extracting serum from an immune animal and injecting the same in non-immune animals. This type of immunity against pathogens or antigens is known as passive immunity as the antibodies grant the recipient a temporary resistance and as they are not born of the recipient's own immune response against the invaders. The contributions of Kitasato and Behring did not go unnoticed. Both scientists were bestowed with titles, where Behring became von Behring and Kitasato became Behring Kitasato. Behring was awarded the first ever Nobel Prize in Physiology or Medicine in 1901 in recognition of his work. The observations of Behring and Kitasato, in conjunction with others, paved the way for the introduction of passive immunization in clinical practice. Passive immunotherapy, a practice by which resistance to pathogens can be transferred from an immunized donor to an unimmunized recipient through the transfer of antibodies, was heavily in the 1930s and 1940s to endow peoples with resistance against hepatitis A and measles. Antiserum, the serum fraction of blood-containing antibodies, were derived generally from horses and made use of. Advances in technology for immunoglobulin preparation and increasing experience has made these procedures quite routine in modern medicine. In cases of immunodeficiency and for some autoimmune diseases, passive immunization therapy is frequently used. This helps in the protection of individuals from anticipated infections and exposure against which they cannot put up any form of resistance. Passive immunization can also be life-saving in episodes of certain infections, such as following a rabies infection. Additionally, antiserums containing antibodies against snake or scorpion venom is also used for bite victims who have suffered such bites. Furthermore, newborn infants also benefit from passive immunity. In this case, maternal antibodies pass into and circulate in the fetus to provide protection against possible pathogens. One of the historical cases of significance here is passive antibody therapy saved many lives occurred in 1925, when an outbreak of diphtheria was detected in the then remote outpost of Nome in Alaska. Antibodies against diphtheria were available in Anchorage, but the weather was at its worst, and neither roads were open nor were flights flying between the two places. The events that followed is a testament to how the sheer will of men can overcome the toughest of challenges in the direst of circumstances. In a nearly impossible attempt to save the lives of several children suffering from diphtheria, a dog sled relay was set up to cover a distance of 700 miles between where the railroad ended in Inanna to Nome. In this relay, two Norwegians covered the most dangerous terrains under blizzard conditions. Leonard Seppala, who covered particularly treacherous terrain, and Gunnar Kaasen, who along with his lead dog Balto, drove the final leg of the relay in whiteout conditions. Kaasen and Balto arrived just in the nick of time to save several children that were suffering from the disease. To commemorate this heroic endeavor, a statue of Balto was erected later that same year in Central Park in New York, where it still stands today. This journey is memorialized every year now through the running of the Iditarod sledge dog race, a nearly 1,000-mile trek from Anchorage to Nome. This race takes 10 to 15 days to complete with mushers leading teams of 14 sledge dogs. As mentioned above, passive immunotherapy provides a quick but temporary solution to infection since the cells that produce the antibody specific for the pathogen or antigen are not transferred between individuals. Vaccines or a natural infection, on the other hand, creates active immunity in the host, which refers to the development of one's own immunity against a pathogen. Active immunity against pathogens provides individuals with a humoral immunity. Metchnikoff's concept of cell-mediated immunity, where the immune response was imparted by specific immune cells, was termed cell-mediated immunity. The relative contributions made by each of these two types of immunity was a matter of raging controversy at that time and was widely debated. Of course, it is quite obvious now that both viewpoints are correct, since a mature immune response requires and employs both the humoral and cell-mediated components of the immune system. A complete understanding of the immune response was hindered primarily by the lack of genetically modified animal models and modern tissue culture methods. The research on cell-mediated immunity and the contributions of immune cells thus lagged behind that of humoral immunity, which marched forward, taking advantage of the ready availability of blood and established protein purification techniques. In the 1940s, Merrill Chase, then working at the Rockefeller Institute, successfully conferred immunity against tuberculosis in guinea pigs through the transfer of white blood cells from an immunized to an unimmunized one. This was a key experiment that helped reinvigorate the interest in cell-mediated immunity since all attempts to develop a vaccine against tuberculosis had met with failure up until that point. With the emergence of improved cell culture techniques in the 1950s along with advances in transfer techniques, lymphocytes were finally recognized as the effectors of both humoral and cell-mediated immunity. Experiments with chicken carried out by Bruce Lick at the Mississippi State University soon thereafter indicated the existence of two types of lymphocytes, T lymphocytes T cells and B lymphocytes B cells. They were named so because T cells were derived from the thymus, and B cells were derived from the bursa of the brisseus in birds. Interestingly, in what can only be termed as a convenient twist of nomenclature, B cells were found to develop in the bone marrow in mammals, thus making it easier to remember in a way. It is now well known that T cells impart cell-mediated immunity and an immune response while B renewable and long-lived protection through memory cells that can remain active for years or even decades depending on its cognate antigen. Much before the discoveries by Behring and Kitasato that serum components can transfer immunity against a pathogen, Elie Metchnikoff, a Nobel Prize winner, had demonstrated in 1883 that certain phagocytic cells contribute to immunity in an animal. He had observed white blood cells, which he named phagocytes, in the act of ingesting foreign substances and microorganisms. He also noted that these phagocytes were more active in animals that were immunized and hypothesized that cells were the major effectors of immunity and not soluble components. The white blood cells observed by Metchnikoff were most likely blood monocytes and neutrophils. The observations of Kitasato and Behring, and also Metchnikoff, gave rise to a long-lasting debate among immunologists that raged on for decades. In the search for the protective agent that confers immunity, several researchers helped identify the active immunological component in blood. This soluble component was being actively characterized by various groups of scientists the world over and was found to be able to neutralize or precipitate toxins and agglutinate clump bacteria. Depending on the context of the finding, this component acquired various names: antitoxin, agglutinin, precipitin, and so on. It was unclear initially whether a single or multiple components of serum were responsible for such activity. However, during the 1930s, mainly due to the efforts of Elvin Cabot, a serum fraction called gamma globulin, now called immunoglobulin, was inferred to be responsible for the observations or activities. Clearly, a clinical practice involving the use of this fraction of the serum that contains these active molecules, which we now know to be antibodies, was expedited, and the basic understanding of the actual mechanism of the transfer of immunity from one person to another lagged behind. Since these antibodies were found in body fluids known at that time as body humors, the immune response orchestrated by them was termed different antibodies. Two major theories were put forward to explain these observations: the selective theory and the instructional theory. The selective theory was founded by Paul Ehrlich in 1900. To explain antibodies found in serum, Ehrlich proposed that blood contains cells that express a diversity of receptors, which he named side chain receptors that could bind pathogenic elements and inactivate the same. Ehrlich proposed that much like the fit between a key and lock, the specific receptors on these cells would bind to specific antigens and no other. Ehrlich's theory was inspired from the lock and key model proposed by Emil Fischer in 1894 to explain the interaction between an enzyme and its substrate. Ehrlich further proposed that when such an interaction between a pathogen and a receptor takes place, the cell will be induced to produce more receptors with the exact same specificity. He thus imagined these effector immune cells to be pluripotent that expressed a number of receptors, each of which can theoretically be selected. Ehrlich explained that the specificity of these receptors was predetermined in the host, i.e. before exposure to the antigen, and therefore it was the antigen that selected the receptor. As we know now, most of Ehrlich's theory has been proven with the one exception that instead of one single cell expressing many receptors, each cell makes multiple copies of only one membrane-bound receptor with the same specificity. Thus, for the immune system to work, an army of immune cells, carrying receptors with a wide variety of specificities is required. B cells that are selected by antigenic interactions are activated, upon which they proliferate and consequently secrete thousands of copies of the soluble form of this receptor antibodies, which attach to pathogens and label them for destruction. Cells are responsible for conferring humoral immunity. The real controversy regarding the roles of humoral immunity, as opposed to cell-mediated immunity, was resolved only when it was shown that both systems were intertwined and that both were necessary for a mature and complete immune response against an antigen or a pathogen. The theory of clonal selection. Another one of the great mysteries that were encountered by early immunologists was what determined the specificity of an immune response to a particular antigen, the general term used to describe any foreign substance which elicits an immune response in T or B lymphocytes. Around 1900, Jules Bordet, working then at the Pasteur Institute, added to the understanding of antigens by demonstrating that non-pathogenic entities such as red blood cells from other species could serve as antigens. It was shown that serum from an animal that has been inoculated with a foreign, non-self, but otherwise non-infectious substance would react with the injected substance in a specific manner. Studies carried out by Karl Landsteiner and others who followed him demonstrated that an animal was capable of developing specific immune responses against almost any non-self organic material through the production of antibodies. These antibodies would expectedly bind specifically to the foreign substances. It was additionally found that antibodies were capable of developing specific immune responses against compounds that had been recently synthesized in the laboratory and were not even found in nature. All this evidence pointed to the seemingly infinite range of reactivity that antibodies possess. The specificity of antibodies was also intensely studied, whereby it was shown that molecules differing in even a single amino acid could be distinguished by their reactivity with. Figure 4.94 Paul Ehrlich's side chain theory for explaining the formation of antibodies. Ehrlich initially proposed the cell to be pluripotent, where it expresses a variety of side chains or receptors, each with different specificities. When an antigen is encountered by this cell and is bound by one of its receptors optimally, the cell is stimulated to produce more receptors with the same specificity, and the receptor is released. The instructional theory was proposed to challenge the selection theory in the 1930s and the 1940s. Several such theories were suggested, and most proposed that antigens played a critical role in determining the specificity of an antibody. Instructional theorists explained that a particular antigen would act as a base or template, around which the antibody would fold, much like an impression or mold. Thus, the antibody would take up a conformation that is complementary to or fitting of the antigenic template, lending it the specificity for that particular antigen. This theory was first proposed by Friedrich Breinel and Felix Haurowitz in 1930, but was further refined and explained in terms of protein folding in the 1940s by Linus Pauling. Selective theories again resurfaced as new experimental data accumulated in the 1950s. Led by insights provided by Niels Jerne, F. McFarlane Burnett, and David Talmage, the selection model was refined to what is now known as the clonal selection theory. Over the years, this hypothesis has been refined further and is now widely accepted as the underlying archetype of modern immunology. The theory of clonal selection proposes that each B and T lymphocytes in an individual express several copies of the same membrane-bound receptor, which share the same, distinct specificity for a particular antigen. The specificity for this unique receptor pertaining to its anti-inhibiting capability is determined before its exposure to the antigen. The cells are activated when the cognate antigen binds to the receptor, and this leads to a proliferation of the cell into a clone of daughter cells that bear the same receptor and specificity as the parent cell. Instructional theories were formally disproved in the 1960s. By this time, information was being generated about the structures of DNA, RNA, and protein. By this time, information was being generated about the structures of DNA, RNA, and proteins that offered novel insights. By this time, information was being generated about the structures of DNA, RNA, and proteins that offered novel insights into how lymphocytes could make antibodies against almost anything. The overview presented in Fig 4.95 presents a relatively diluted scheme for clonal selection as observed in the humoral B cell and cellular T cell branches of immunity. It is now known that B cells produce antibodies, a soluble version of their antigen receptor molecules that bind to antigens and flag them for destruction. T cells, which can be of different types, as discussed later in this chapter, also use their surface-bound antigen receptor molecules to bind antigens. Once selected by antigen binding, T cells can perform a number of functions which include the secretion of signal molecules that aid in the activation, proliferation, and the differentiation of other immune cells such as B cells and destruction of infected host cells. Figure 4.95 Humoral and cellular immunity. The humoral branch of immunity involves the interaction of B cells with foreign proteins referred to as antigens, followed by differentiation into antibody-secreting immune cells. Once secreted, antibodies bind to these antigens and help in flushing the body of infectious agents. Cell-mediated immune response involves diverse subsets of T lymphocytes, which are capable of carrying out various immune functions, including secretion of soluble molecules which stimulate other immune cells and direct killing of infected cells. Overview of the mammalian immune response. The mammalian immune system is a highly evolved biological machine that we are only beginning to understand at the cellular and molecular levels. As evident from the historical skin and mucous membranes. Conditions such as the acidic pH of the stomach, the vagina, and of sweat also act as barriers to microbial growth, even though not truly physical barriers. The immense importance of these generally unappreciated and less glamorous defenses of the human body is often only understood when these are surmounted. For instance, puncture wounds of the skin can transmit diseases such as plague, fleet, malaria, mosquitoes, and Lyme disease ticks, while animal bites can convey diseases such as rabies and tetanus. An extreme example of the utility of these obvious defenses is seen in burn victims, where protection provided by the skin is lost at burn sites and therefore mandates aggressive therapy with antibiotics to prevent or combat the unbridled microbial infections that often follow. The immune system adapts readily to the pathogen. The immune response, as detailed in the previous section, is thus highly inspired by the type of pathogen it encounters and how and where it does so. To elaborate, chemical structures found on the pathogen and whether the pathogen is encountered in an extracellular or an intracellular environment are some factors that determine the population of cells and molecules of the immune system that respond to the invasion. The most effective way to deal with the invading pathogen is decoded by the immune system through the evaluation of different chemical structures and microenvironmental cues. Pathogens are recognized by the immune system through interactions between the foreign organism and recognition molecules expressed by the host. Such recognition molecules may be of two primary types: membrane-bound receptors or soluble receptors, secreted molecules. The cognate ligands for these host receptors include secretions of the foreign organisms, whole cells of the pathogen, and antigens derived from the pathogen. These interactions lead to an intracellular or extracellular chain of events that eventually results in the labeling and consequent destruction of the foreign organism. Studies described above, the immune system is a complicated network of cells and soluble agents that has matured to provide protection from an enormous range of pathogens of all forms, shapes, and sizes. In this section, we discuss the unique and salient characteristics of the immune system and attempt to understand the mammalian immune response as an overview. Natural barriers form the first line of defense against pathogens. Pathogens are organisms that are causative agents of diseases, and the process through which they induce sickness in hosts is called pathogenesis. Most human pathogens are one of four types: viruses, bacteria, fungi, and parasites. The immune system exploits inherent differences between the host and groups of pathogens to its own advantage to recognize and destroy invaders. The microenvironment in which the pathogen is encountered by the immune system plays an important role in how it responds to it. The same pathogen may elicit very different responses depending on the microenvironment where it was encountered. For example, certain areas of the human body are considered too sensitive and are off-limits for the immune system. This includes the central nervous system, where the immune system can potentially do more damage than a pathogen. In contrast, there are other areas of the body which have well-defined strategies for the immune system. One such example is the digestive tract, where copious amounts of foreign substances enter regularly, including commensal microbes that help us digest our food, but are tolerated by our immune system. However, when these same microbes enter the bloodstream, they are treated much more aggressively by the immune system. Thus, the immune response broadly varies not only depending on the type of pathogen and on the microenvironment wherein it encounters the pathogen. Immune response, however, is not initiated until the physical barriers of the body are breached, and the pathogen truly enters the body. Obvious physical barriers include that this is more simply described as an immune response. This includes the involvement of a complicated network of cells that are able to recognize and ill or engulf a pathogen, cellular immunity, as well as soluble proteins that facilitate the labeling and destruction of the invading organisms, humoral immunity. Depending on the type and scale of recognition molecules engaged, the nature of the immune response can vary greatly. An example can be given of a viral attack on the human body. Viruses are tiny, obligately intracellular organisms that spend most of their life cycle inside host cells. An effective approach to defending against viruses, therefore, must include the identification of the pathogen through its surface as well as recognition of infected cells. A subpopulation of the immune cells must be able to identify changes in a host cell once it is infected and forward the immune response. This function is achieved by cytotoxic T lymphocytes, which are a vital part of cellular immunity. In case of virus-infected cells, recognition molecules inside the cell are crucial to the initial immune response. These intracellular receptors bind to viral proteins inside the cell and alert the cell to the presence of an invader through the initiation of an early warning system. Often, the only true way of eradicating a viral invasion involves the destruction of the host cell. Generally, such a strategy is effective for the host, but in certain instances, it can cause severe impediments. For example, the human immunodeficiency virus (HIV) infects a subpopulation of T lymphocytes called T helper cells. These cells are named due to the facilitated role in a holistic immune response, whereby they can direct the behavior of B cells and are therefore crucial to a successful immune response. Upon destruction of a large enough number of cells or upon rendering many such T helper cells dysfunctional, the directional cues required for a complete and healthy immune response are absent and combating further infections becomes difficult. As such, immunocompromised patients often suffer from one or more opportunistic infections and frequently succumb to them. In case of extracellular pathogens such as fungi, most bacteria, and some parasites, the immune system employs drastically different strategies. Such immune responses are primarily dependent on the recognition of foreign organisms through cell surface receptors or soluble molecules that routinely scan the extracellular spaces of the body. Thus, in this case, B cell receptors on the surface of B cells and antibodies secreted by them, which are a part of humoral immunity, play a pivotal role. Antibodies, for example, can reach spaces in the body where B cells themselves may not be able to reach and help identify pathogens in such remote locations. In the case of large parasites, their size restricts them from being engulfed by phagocytes. Under such conditions, cells that are able to deposit toxic products on the parasite or secrete products that can induce expulsion, e.g., coughing, vomiting, sneezing, are apt strategies. With the subsequent discussions, it will be increasingly evident that a one-glove-fits-all approach does not exist for immune responses. The variations in pathogens and their pathogenesis mandates a great diversity in the solutions to combat them. At the same time, the different pathways of the immune system do not function in a discrete manner and exhibit considerable overlap in structure and function. Pathogen recognition molecules can be either germline encoded or randomly generated. It is fairly easy to understand that pathogenic microbes invading the host will bear some unique differences with it. These chemical structures, which are not typically found in mammals, have been collectively called pathogen-associated molecular patterns, or PAMPs. PAMPs can be defined as foreign structures that are common to groups of pathogens and are characteristic of them. The mammalian immune system recognizes these foreign structures at the very start of an immune response. Both vertebrate and invertebrate organisms have immune systems which have evolved to give rise to cell surface receptors and soluble proteins that can promptly recognize most of these PAMPs. Such pathogen profiling is immensely important to launch the first wave of the immune response. For instance, encapsulated bacteria contain a specific polysaccharide with a unique structure in their cell surface coat which is not found in either mammalian cells or other bacteria. These sugar residues are recognized by some of a diverse array of receptors, collectively called pattern recognition receptors (PRRs), alongside other such common foreign structures. Upon detection of these alien structures by the PRRs, a train of events label the pathogen for destruction. PRRs are encoded in the genomic DNA and are therefore always expressed by the various immune cells. These germline encoded recognition molecules constitute the first line of defense of the immune system and aid in the quick detection of several typical foreign identifiers found in most pathogens. An important consequence of the presence of PRRs is that the engagement of these molecules allows the early profiling and categorization of the type of pathogen that is of immediate concern. This understanding is crucial for the subsequent steps in the immune response where the preferred immune response routes are to be activated along with further fine-tuning of the immune response as it continuously develops. As an instance, viruses expose unique chemical structures inside the host cells only during the replication stage in their life cycle. Several of these chemical structures are recognized by intracellular receptor molecules and are subsequently bound by them. This in turn induces an immediate antiviral response in the infected cell, which halts further replication. Simultaneously, this event initiates the secretion of signal molecules that act as warnings for neighboring cells to aid in the prevention against infection. The early profiling and categorization of the pathogen allows the immune system to record the recognition molecules involved in the initial detection event and subsequently convey the relevant information to effector immune cells. This allows an increasingly focused immune response specific for the type of pathogen encountered. The host and the invading pathogen are always locked in a tussle for dominance. Pathogens evolve to produce unique signatures that are capable of avoiding detection by the host, and the host germline encoded PRRs also evolve to rise up to these new challenges. However, because pathogens generally have much shorter lifespans than their animal hosts and also use error-prone DNA polymerases for replication of their genomes, they can evolve much faster than their hosts to facilitate evasion of the immune system. It is understandable, therefore, that if the PRR-based immune response was the only attack a host could launch on an incoming pathogen, it will soon become obsolete. A system of immune response thus is necessary that involves the ability to foresee possible foreign chemical structures and add diversity to the PRR mechanisms to negate the random nature of pathogenic structures that develop over generations of the invader. To this end, the vertebrate immune system employs a resource-intensive and complicated system based on the allowance of randomness in the design and generation of certain recognition molecules. Such a strategy, whereby diversity is generated concerning the recognition of alien pathogens, is employed only in B and T lymphocytes. As a result, this process gives rise to the population of B and T lymphocytes, each of which expresses a single, unique recognition molecule targeted at a specific structure. In their entirety and with the randomness involved in the generation of the binding sites for these recognition molecules, these cells have a theoretical potential to respond to any incoming antigen. Such an undertaking is accomplished by the rearrangement and editing of the genomic DNA that encodes the antigen receptors for each B and T lymphocyte. Similar to the error-prone DNA replication mechanisms that can be attributed to microbial pathogens, chance plays a large role in the said process to generate an array of diverse recognition molecules. The editing of the chromosomal DNA, cutting and splicing, however, has certain risks. To alleviate them, these lymphocytes must survive the DNA editing procedures and, more importantly, the quality control processes that follow it. These procedures take place in the primary lymphoid organ of maturation for these lymphocytes, thymus for T cells and bone marrow for B cells. The surviving population of cells move into bodily circulation and scan bodily spaces for their cognate antigens. If and when the cognate antigen is encountered by these surface receptors on these cells, a clonal selection of these cells is initiated. The selected clone undergoes rapid proliferation to create thousands of copies of itself, all bearing the same recognition molecules. This leads to an increase in the binding of the particular antigen and eventual labeling and destruction of the invading pathogen. In T lymphocytes, the recognition molecule is a membrane-bound T cell receptor. In B cells, these recognition molecules can have two forms, a membrane-bound receptor form and a secreted antibody form. The molecular mechanisms behind the generation of B cell receptor and antibody diversity were discovered by Susumu Tonegawa at the Basel Institute for Immunology in Switzerland in the year 1976. The discovery of these mechanisms marks a true turning point in the understanding of immunology, a coming of age of sorts. It was understandably one of the most significant discoveries in the history of immunology and received widespread recognition, with Tonegawa being awarded the Nobel Prize in Physiology or Medicine in 1987. Tolerance and non-self discrimination keeps the immune system from destroying the host. The principal problem in the allowance of randomness in the generation of recognition molecules or receptors for pathogens is that some of these receptors can recognize host structures and not foreign ones. So, for this system and the immune system at large to function properly, cells which recognize host structures must be kept from accidentally recognizing and consequently destroying host tissue. This involves the principle of non-self discrimination and is simply called tolerance. Another hallmark of the vertebrate immune response, the discovery of the presence of such an element in our immune systems, is credited to F. McFarland Burnett and Peter Medawar. Burnett proposed that exposure of vertebrates to antigens at certain specific periods of their life can make their immune system tolerant to these antigenic structures later. The validity of this theory was later proven by Medawar, who exposed mouse embryos to specific foreign antigens and showed that these mice were able to tolerate said antigens later in life. Both Burnett and Medawar were awarded the Nobel Prize in Physiology or Medicine in 1960 for their work. For establishment of tolerance, the recognition molecules present on the B and T lymphocytes generated through the random DNA editing process must first pass a test of non-responsiveness against host structures. Shortly after the generation of these receptors, the process to test these receptors against host structures start, and upon identification of cells with receptors that recognize the host, they are killed or inhibited. The proper identification and consequent destruction of B and T cells with inadvertently generated receptors that recognize the host is key to the successful maintenance of tolerance and, in turn, non-self discrimination. Recent reimaginings of the concept of tolerance involves the danger hypothesis. Proposed by Polly Matzinger in 1994, the danger hypothesis suggests that the immune system constantly determines each new encounter with a foreign structure more for its potential to be harmful to the host rather than whether it is self or not. For example, a certain cell can die due to a number of reasons which include natural causes, infection or mechanical damage. While the former is a natural consequence of the aging of a cell and requires a regular cleanup of the cellular debris, the later two instances are not natural and are associated with the release of intracellular contents, pathogen-specific products and expression of cellular stress proteins. These cell stress or pathogen-associated cues, collectively called danger signals, have the ability to engage specific host recognition molecules that recruit immune cells to get involved in unnatural causes of cellular death. Although a stringent and robust self-tolerance system protects the vertebrate body from unintended harm that can be caused by the immune system, it also helps cancer cells avoid detection as long as they express host cell structures that the immune system has been trained to ignore. Most autoimmune diseases are a direct consequence of an abnormal tolerance of the host immune system. This is understandable, and often such failure to maintain or establish tolerance can have a devastating effect on the host. Innate and adaptive immunity. Up to this point, the immune system has largely been referred to as a complicated, singular system. It is important to understand that it really consists of two reasonably interconnected systems of immunity, innate and adaptive. Both of these systems are required and collaborate to combat the pathogens that invade the vertebrate bodies. Innate immunity can be defined as all genome-encoded molecular and cellular mechanisms and are evolutionarily primitive in nature, where the main objective is to promptly eliminate pathogens or to prevent infections altogether. This may include physical or chemical barriers to the entry of pathogens, and germline-encoded receptors for recognition of frequently encountered pathogenic chemical signatures such as PRRs. The outcome of an innate immune response amounts to prompt recognition of the pathogen followed by phagocytosis or destruction of the same. Another important component of innate immunity includes a series of pre-existing serum proteins, collectively called the complement system. The complement system binds most camps and labels them for consequent destruction. This highly efficient and effective system prevents an incoming pathogen to gain ground and eliminates the invader within hours of the encounter. Recognition elements of the innate immune system facilitate fast identification times, but are not very specific and are unable to differentiate between antigens with minute differences. The innate arm of immune responses thus is designed to identify, attack and tackle growth signatures among pathogens and can be equated to hammer. The second arm of the immune system, known as adaptive immunity, is capable of recognizing subtle molecular differences in foreign structures and is responsible for much more nuanced and focused immune responses. This arm of the immune system relies on the B and T lymphocytes and exploits their ability to identify a large pool of antigenic structures. Adaptive immune response is typically much slower than innate responses but are much more antigen specific. Such a response against a pathogen can take up to 5 to 6 days to surface after the initial contact or breach of barrier and is maintained until the infection is resolved. There are two principal reasons for the slower emergence of an adaptive immune response. Firstly, very few TRB lymphocytes possess the exact, randomly generated, the cognate receptor that can bind the relevant antigens, and until the initial binding event happens, clonal selection cannot start. Secondly, parts of the adaptive immune response also relies on cues and signals generated from prior encounters from the innate processes to identify the type of antigen. Upon encountering their cognate antigen, the T and B lymphocytes undergo clonal selection and proliferation. Even though the adaptive immune response is slow, once selected, B and T cells put up a formidable defense against almost any invading pathogen that mostly ends in the proper resolution of the infection. The adaptive arm of immune responses thus is designed to identify, attack and tackle much more subtle signatures among pathogens as compared to the innate arm and can be equated to scalpel. Unlike innate immunity, the adaptive arm of an immune response evolves in real time in response to an infection. It continually adapts to better recognize, eliminate and memorize the invading pathogen. Adaptive immune responses are inherently complex and involve an interwoven mix of cells and molecules that work together to resolve a job initiated during the innate immune response. Vaccines use the ability of adaptive immune responses to generate memory, whereby vaccinations aim at the development of a specific and long-standing adaptive response that can garner protection to the vaccinated individual in the future in case of a real infection. As must be increasingly clear by now, the adaptive arm of immunity relies heavily on the randomly generated but highly specific receptors carried on B and T lymphocytes leading to the manifestation of a multifaceted and robust immunity. The complexities and the sophistication attributed to the adaptive immune system must not be taken as a slight on the innate immunity arm of the immune system. Neither is the adaptive arm of the immune system more important than the innate, nor is it independent of it. The maturity of an adaptive immune response, in fact, is highly dependent on the earlier innate immune pathways. The intertwinings and intricacies of the connections between these two arms remain a subject of intense research. In 2011, Bruce Butler and Jules Hoffman were awarded the Nobel Prize in Physiology or Medicine for their discoveries pertaining to activation events important for innate immunity. The same prize was also shared by Ralph Steinman, who was rewarded for his discovery of the role of dendritic cells in the activation of adaptive immune responses. Since the innate immune pathways encounter pathogens first, the cells and molecules of this branch of immune response relay the information gathered during their early encounters with the pathogen to help direct the adaptive immune response. As mentioned before, adaptive immune response, therefore, provides a second and more comprehensive defense of the vertebrate body, informed and aided by the confrontations borne by the innate system. Some infections which are fairly localized and involve reasonably low numbers of benign foreign invaders are taken care of completely by the innate immune mechanisms alone. Principal features of innate and adaptive immunity are tabulated in tab 4.17. The two arms of the vertebrate immune mechanisms are typically elaborated separately for the ease of discussion, but incorporate considerable overlap of the cells and molecules involved. Features innate immunity, adaptive immunity. Response time. Minutes to hours, days. Major components: physical barriers such as skin, pattern recognition molecules, phagocytes, B and T lymphocytes, antigen specific. Specificity limited, highly specific and diverse, adapts during the course of the infection, thereby increasing specificity further. Response to repeated infection. Same response each time, increasingly rapid and effective with every infection. Table 4.17 features of innate and adaptive immunity. Convergence between the adaptive and innate arms of the immune system requires robust communication mechanisms between the two. This communication is achieved through the use of both cell-cell contact and soluble messengers. Most soluble messenger molecules that are involved in such communications are growth factor-like molecules collectively known as cytokines. Binding of these cytokines and other cell surface ligands to receptors on effector cells signal these cells to perform different new actions, such as the synthesis and release of other soluble molecules or differentiation to new cell types. A subpopulation of these signaling molecules are called chemokines, as they have the ability to attract specific cells to a site, i.e. have chemotactic activity. Soluble factors, including cytokines and chemokines, direct the immune response and recruit and instruct immune cells from the infection site in this way. This recruitment can be felt in the form of swelling, heat, and tenderness at the site of infection and are part of a larger process collectively referred to as an inflammatory response. Generation of memory is a seminal feature of the adaptive immune response. One of the most important and unique characteristics of the adaptive response of the vertebrate immune system is immunologic memory. Immunologic memory grants the immune system the ability to respond to a second exposure to the same pathogen with heightened swiftness and greater efficiency. Unlike other biological systems, the vertebrate immune system is unique and fascinating in their ability to not only adapt to foreign antigens in real time but also remember this information for future use. When a foreign antigen is encountered by the immune system for the first time, the adaptive arm goes through a primary response to the pathogen. During the primary response, cognate lymphocytes relevant to the infection and pathogen are clonally selected, proliferated and finally used for eradication of the pathogen. These cells also receive signals from the innate arm of the immune response to help further fine-tune the response to a specific pathogen. Each and every subsequent encounter of the immune system with the same pathogen is referred to as a secondary response, Fig 4.96. During a secondary response, memory cells which were generated from the B and T lymphocytes deemed most effective against the pathogen during the primary response are enlisted to initiate the attack. These cells start from where they ended during the primary response and proliferate rapidly while continuing to adapt in better strategies for the eradication of the pathogen during each encounter with the antigen. Memory cells can live for decades depending on the antigen under consideration. These memory lymphocytes are key to secondary responses that are so prompt, antigen-specific and effective that upon infection of the host by a pathogen a second time, the malady is often resolved without the development of any symptoms. This remarkable attribute of the immune system to remember their adversaries keeps us from getting infected by the same pathogen again and again. The immunologic memory harbored by B and T lymphocytes also forms the basis of vaccination, where memory against real pathogens are elicited through the use of dead or attenuated forms of the pathogen, i.e. a vaccine, and prepares the immune system for a possible later attack by the actual, life-threatening pathogen. Figure 4.96 differences in the primary and secondary response to antigen exhibiting the phenomenon of immunologic memory. When injected with an antigen, an animal produces a primary antibody in low proportions and short duration, peaking at 10 to 20 days. A second exposure with the same antigen results in a secondary response which is much greater in magnitude, much more specific to the antigen, and peaks in significantly less time, 1-4 days. Innate responses have no memory element, and their immune response remains unchanged regardless of the frequency at which they encounter antigens. An overview of a typical immune response and how the innate and adaptive arms of the immune system work together to inhibit a pathogen. As can be seen in the figure, Fig 4.97, an infection is initiated when a pathogen breaches the skin, mucosal barrier or mucosal lining of the throat. Once through the breach, the pathogen is recognized and engulfed by phagocytic cells. These phagocytic cells, being a part of the innate immune response, release chemokines and cytokines that recruit other white blood cells to the site of exposure, thus initiating inflammation. Phagocytic cells can then travel to the local lymph node, where it facilitates interactions between the pathogenic antigens and resident B and T lymphocytes. Lymphocytes with receptors that are specific for the antigen under consideration are then selected, activated and initiate the adaptive immune response through rapid proliferation. Activate T helper cells facilitate the activation of B cells, and these lymphocytes undergo clonal expansion in the lymph node. Consequently, an army of T and B lymphocytes that are highly specific for the antigen are created with the B cells secreting antibodies that attach to the pathogen and direct its destruction. Finally, this adaptive response leaves behind a posse of memory B and T cells available for secondary encounters with the same antigen that may take place in the future. It must be noted that immunologic memory is a unique attribute of the adaptive arm of the immune response. Innate immune responses typically do not generate any immunologic memory. In the case of certain vaccines, a single round of vaccination is not enough to elicit an adaptation that can provide adequate protective immunity from the pathogen in question. In several of these cases, such long-lasting immunity only develops after a second or even third round of antigen exposure. Such pathogens require one or more booster shots for the complete development of protective immunity against it. Booster shots are nothing but additional rounds of exposure to the same antigen that further drives and hones the adaptive events required for the development of a final and effective secondary immune response. The objective lies in the refinement of these adaptive responses to such a level as to ensure complete protection against the real pathogen in future. Figure 4.97 Coordination between innate and adaptive immunity. Markings in the diagram represent the following. 1. Entry of pathogen. 2a. Phagocytes carry antigens to local lymph nodes. 2b. Resident phagocytes release cytokines at the site of infection to recruit other immune cells. 3. T and B cells in the lymph node with cognate receptors to the antigen undergo clonal selection, thereby starting adaptive response. 4. T and B cells proliferate and differentiate into effector immune cells such as 5a. Antibody secreting B cells. 5b. Memory T and B cells which aid in rapid responses in case of a secondary infection with the same antigen at a later stage. Origin and maturation of immune cells and effective immune response against external pathogens. Antigens depends on finely tuned and extensively choreographed interactions among the various players or cell types. In general, the first line of defense against pathogens is mounted by cells of the innate immune system, followed by antigen presentation by antigen-presenting cells to stimulated lymphoid cells, which in turn coordinate the adaptive immune response to generate antibodies and memory cells, which prevents future infections. The microanatomy and the specialized, functional anatomy of the immune systems allow it to unleash a coordinated, full-scale immune response against foreign invaders of the body. Dispersed throughout the body, the immune system organizes its diverse cell types in time and space to maximize its effectiveness. Two major components of the anatomy of the immune system are the primary and secondary lymphoid organs. The bone marrow and the thymus constitute the primary lymphoid organs and are responsible for the development and maturation of immune cells from their immature precursors. The secondary lymphoid organs include the spleen, lymph nodes, and the certain sites in the gut and other mucosal tissues. These are responsible for the exposure of antigens to antigen-specific lymphocytes and coordinating their development into effector and memory cells. Finally, all primary and secondary lymphoid organs are connected through blood and lymphatic vessels, which enable the holistic functioning of these systems. Extraordinarily, all functionally distinctive, mature blood cells, including erythrocytes, lymphocytes, macrophages, and dendritic cells, emerge from a single type of cell called the hematopoietic stem cell (HSC). The process of differentiation by which HSCs give rise to mature blood cells is known as hematopoiesis. Fig. 4.98. As mentioned above, the primary lymphoid organs are responsible for the development and maturation of HSCs into mature immune cells. The bone marrow houses the HSCs, which give rise to all cell types of the immune systems through hematopoiesis, while T cells undergo maturation through thymopoiesis in the thymus. Below we discuss the processes of hematopoiesis and thymopoiesis in brief and get acquainted with the different cells of the immune system. Figure 4.98 Hematopoiesis. Pluripotent hematopoietic stem cells differentiate into myeloid and lymphoid progenitors. Majority of immune cells mature in the bone marrow and subsequently move to peripheral organs via blood. Some immune cells, such as mast cells and macrophages, however, undergo further maturation outside the bone marrow. T cells undergo maturation in the thymus. All immune cells originate from hematopoietic stem cells. Stem cells are specialized cells which have two distinctive features. 1. Stem cells can differentiate into a variety of different cell types. 2. They can regenerate or self-renew. Embryonic stem cells have the capability to differentiate into any type of specialized cell type in an organism. This type of stem cell is known as a pluripotent stem cell. Adult stem cells, in contrast, are multipotent stem cells, which can give rise to more than a single type of tissue-specific cell. Many of the tissues of the human body house adult stem cells that can give rise to a diverse array of cells defining a particular tissue. The HSCs are considered a highly representative adult, multipotent stem cells on account of its capability to differentiate into all types of blood cells. Hematopoietic stem cells constitute a rare subpopulation of the cells in the bone marrow. It is estimated that less than 1 HSC is encountered in a population of 5x10^4 cells in the bone marrow. Additionally, their numbers are strictly regulated through a highly controlled regimen of cell differentiation, cell division, and cell death. Under the steady state or homeostatic conditions, when the organism is not under any kind of stress and the immune system is largely idle, most HSCs are quiescent. Few HSCs divide under these conditions to form daughter cells, some of which retain the ability of the mother stem cells, the ability of self-renewal, and the capability to give rise to all blood cell types. Daughter cells that do not differentiate into stem cells evolve into progenitor cells, which progressively commit to a particular blood cell lineage. Importantly, it must be understood that the HSCs have a limited capacity for self-renewal. This is evident from the fact that as the organism ages, the number of HSCs gradually decreases. The extensive proliferative capacity of the HSCs is displayed when there is an increased demand for hematopoiesis. During events such as an infection or chemotherapy, HSCs have been shown to divide at much higher rates than under homeostatic conditions. Experiments in mice whose hematopoietic systems were completely destroyed through irradiation showed that the infusion of a fraction of the bone marrow cells could rejuvenate the subject's immune systems. In brief, a lethal dose of 950 rads of X-rays was used to obliterate the hematopoietic system in mice. Such mice generally die within 7 to 10 days of irradiation unless infused with fresh bone marrow cells from genetically identical donor mice. A healthy, normal mouse has approximately 3x10^8 bone marrow cells. Astonishingly, it was observed that the infusion of 104 to 105 bone marrow cells from a donor was enough to completely restore the hematopoietic system. This kind of recovery is a testament to the enormous self-renewal capacities of the HSCs. Over the years, our ability to identify and purify HSCs has improved considerably. Presently, mice irradiated by lethal doses of radiation can be rescued by the infusion of only a few purified HSCs, which directly give rise to progenitor cells that quickly repopulate the blood system. Hematopoiesis, from HSCs to mature blood cells. A hematopoietic stem cell that is induced to undergo hematopoiesis or differentiation loses its capacity for self-renewal and commits itself to one of two broad lineages. It can differentiate into a common myeloid erythroid progenitor, CMP, which is the progenitor cell for all red blood cells, the erythroid lineage monocytes, granulocytes, and macrophages, the myeloid lineage, or it can become a common lymphoid progenitor, CLP, which gives rise to T lymphocytes, B lymphocytes, and NK cells. NK cells and myeloid cells are part of the innate immunity and constitute the first responders to an infection or other trauma. Lymphocytes, on the other hand, are members of the adaptive arm of an immune response and generate an antigen-specific immune response that also gives rise to immune memory. As HSCs progressively mature through these lineages, they lose the capacity to contribute to other cellular lineages. Interestingly, both myeloid and lymphoid lineages can give rise to dendritic cells, which are specialized antigen-presenting cells with diverse functions and features and are crucial to the initiation of the adaptive immune response. The progression of an HSC towards increasing commitment to a particular cell lineage is coupled with genetic changes. Several genes that specify lineage commitment have been identified. Unsurprisingly, many of these identified genes are transcriptional regulators. For example, the transcription factor GATA2 is crucial to the development of all lineages in hematopoiesis. Animals died in the absence of GATA2 during embryogenesis. BMI1 is another transcriptional regulator, which is required for fulfilling the self-renewal capacity of HSCs, and absence of BMI1 in animals leads to their death within 2 months of birth since they fail to repopulate their red and white blood cells. Other transcription factors such as Icaros and Notch have been identified to have much more specific effects on the process of hematopoiesis. The transcriptional regulator Icaros is required for the development of the myeloid lineage but not the lymphoid one. Consequently, animals survive in its absence, but are severely immunocompromised and are unable to mount a full immune response. On the other hand, Notch1, one of the 4 Notch family members, is responsible for the regulation of choice between the T and B lymphocyte lineages. With further research, several more transcription factors and master regulators of lineage commitment that are important for immunological development continue to be identified. Environmental changes experienced by an organism can also greatly affect the rate at which hematopoiesis occurs in the production and release of specific cells from various lineages. For instance, it has been seen that cytokines released during an infection can lead to an upregulation in the development of myeloid cells, including neutrophils. Recent evidence also suggests that the release of mature cells from the bone marrow can be responsive to the circadian rhythm and thus be regulated by the sympathetic nervous system. Early immunologists classified immune cells on the basis of their appearance under a microscope. These original classifications were generally carried out with the help of dyes. Such use of dyes was fundamentally helpful in establishing distinctions between myeloid and lymphoid lineages, granulocytes and macrophages, neutrophils, basophils and eosinophils. Stains such as hematoxylin and eosin red H and E are frequently used even today for the distinguish between cell types in blood smears and tissues. Because of their inherent differences in the pH sensitivity and affinity for macromolecules in a cell, these dyes are helpful in highlighting intracellular differences. Thus, a basic dye such as hematoxylin binds basophilic nucleic acids to dye these blue, while the acidic dye eosin binds to eosinophilic proteins found in granules in the cytosol, imparting a pink color. Such stained immune cells and the behavior of live cells in solution were instrumental in helping microscopists deduce the function of these cells. Further advancements, including flow cytometry, greatly impacted our understanding of cell subtypes through the evaluation of multiple surfaces and internal proteins expressed by individual cells simultaneously. Temporal and spatial dissection of the immune response became possible with the advent of sophisticated fluorescent microscopy techniques that allow the observation of live cells in vitro and in vivo. Coupled with genetic manipulation of cell function, these advancements have helped investigators reveal remarkable diversity among myeloid and lymphoid cells and continue to do so. Cells with novel functions are still being discovered, and unexpected relationships between hematopoietic cells are being inferred. Our understanding of the cell subtypes of the hematopoietic lineages is not complete, but an impressive repertoire of knowledge regarding the same has been accumulated over the years. An adaptive immune response is mediated by the cells of the lymphoid lineage. Lymphocytes represent the principal cellular players in the adaptive immune response and can constitute almost 99% of the cells in the lymph. Often comprising 20–40% of the white blood cells circulating in the blood, lymphocytes can be broadly classified into three subpopulations based on functional and morphological differences: B lymphocytes, B cells, T lymphocytes, T cells, and natural killer NK cells. At a time, almost 10–12 trillion lymphocytes circulate the connective tissues in the human body, including the blood and lymph, and migrate into lymphoid organs and tissue spaces. Lymphocytes constitute a relatively morphologically homogeneous population and are quite difficult to distinguish phenotypically. Indeed, the T and B cells appear to be quite similar under the microscope. The distinction between lymphocytes is therefore heavily reliant on the identification of the signature of surface proteins expressed in different types of lymphocytes. Surface proteins expressed in immune cells are commonly referred to as a cluster of differentiation, sometimes also as a cluster of designation, or simply as a CD. This nomenclature was established in 1982 at the first International Workshop and Conference on Human Leukocyte Differentiation Antigens held in Paris, in recognition of the observation that most antibodies generated largely due to the advent of modern monoclonal antibody production technology targeted at surface molecules of human leukocytes were binding to the same protein or antigen. Proposed surface proteins that were bound by two or more antibodies were given the CD nomenclature to differentiate between groups. Although the CD nomenclature was originally designed to identify specific antibodies against human leukocytes, it has now been adopted to describe and differentiate a diverse array of surface proteins on multiple cell types. Table 4.18 lists some of the important CD markers found on human and mouse lymphocytes. CD designation functions: T helper cell Th, cytotoxic, T cell TC, B cell NK cell, CD2 signal transduction, adhesion molecule, plus. CD3 T cell receptor TCR, signal transduction, element, plus. CD4 adhesion molecule which binds to class 2 MHC molecules, signal transduction, plus. Normally, CD5 unknown plus, subset of B cells, plus. CD8 adhesion molecule which binds to class IMHC molecules, signal transduction, normally, variably present. CD designation functions: T helper cell Th, cytotoxic, T cell TC, B cell NK cell, CD16 Fc gamma RIII, low affinity receptor for Fc region of Ig, molecules, plus. CD19 CD21 co-receptor, signal transduction, plus. CD21 CR2 C3d complement receptor, also acts as a receptor for Epstein-Barr virus, plus. CD28 receptor for costimulatory B7 molecule found on antigen presenting cells, plus. CD32 Fc gamma receptor for Fc region of Ig, molecules, plus. Apart from the CD marker proteins, B cells and T cells also express an antigen-specific receptor on their surface, simply called the B cell receptor BCR and T cell receptor TCR. A remarkable diversity of antigenic specificities is covered by B cells and T cells, approximated to be more than a billion. Apart from the CD marker proteins, B cells and T cells also express an antigen-specific receptor on their surface, simply called the B cell receptor BCR and T cell receptor TCR. A remarkable diversity of antigenic specificities is covered by B cells and T cells, approximated to be more than a billion. However, all the BCR and TCR expressed on any individual B cell or T cell is identical in structure and therefore have circulate the human body identical antigenic specificities. As an extension, if a lymphocyte divides to form two daughter cells, antigen-specific receptors expressed in both will have the identical to each other as well as to the parental cell. This holds true for all the descendants these daughter lymphocytes produce too. Thus, a population of lymphocytes originating from a single founding lymphocyte have identical antigenic specificities and therefore is effectively a clonal population. At a given time, several thousand B cell and T cell clones, characterized by their unique and identical antigenic specificities, circulate in the human body. Mature B and T cells are ready for mounting adaptive immune responses upon challenge with an antigen, but until then, they are considered naive. Upon antigenic contact, naive lymphocytes proliferate and differentiate into effector and memory cells. The effector cells function to combat the ongoing invasion, while the memory cells remain quiescent in the host end. Upon a secondary challenge with the same antigen, mount an immune response that is both quicker and greater in scale. In immunological parlance, the first antigenic encounter is termed a primary response, and the re-encounter is called a secondary response. T lymphocytes. T cells derive their designated letter from their site of maturation, i.e., the thymus. Each T cell expresses a unique antigen binding receptor which is simply called the T cell receptor. T cell receptors can only recognize antigens in a processed form, typically in the form of peptides, bound to cell surface proteins called the major histocompatibility complex (MHC) molecules. T cell receptors cannot bind soluble or particulate that is not presented in association with MHC molecules. MHC molecules are genetically diverse glycoprotein molecules that are found on the cell membrane and, depending on the cell, can be of varying types. Cells process internal proteins, both foreign and self, and present them to browsing T cells by binding these processed antigens to MHC molecules and expressing the conjugates on their cell surface. MHC molecules principally belong to two categories: Class 1 MHC molecules, which are expressed in all nucleated cells of vertebrates, and Class 2 MHC molecules, which are expressed by professional antigen-presenting cells and some other cell types during incidences of inflammation. T lymphocytes are primarily of two major types: T helper cells Th and T cytotoxic cells TC cells. These two subpopulations of T cells can be distinguished from each by the presence of either CD4 or CD8 membrane glycoproteins on their surface. As a general rule, T cells displaying CD8 function as TC cells and recognize antigens presented in complex with MHC class 1 molecules, whereas T cells displaying CD4 function as TH cells and recognize antigens presented only in association with MHC class 2 molecules. It has been seen that in normal peripheral human and mouse blood, the ratio of CD4 plus to CD8 plus T cells is approximately 2:1. A change in this ratio is usually indicative of a disorder such as an immunodeficiency disease, i.e., HIV, or other maladies that can lead to loss of immunocompetence. Naive CD8 plus T cells continually browse the surface of antigen-presenting cells, primarily nucleated cells of the body, with their T cell receptors. Upon recognition of a cognate antigen presented in association with MHC class 1 molecules, the naive CD8 plus cells become activated and proliferate and differentiate into an effector cell called a cytotoxic T lymphocyte (CTL). CTLs scour the body, monitoring cells in search for any cells that display foreign antigenic peptides complex with MHC class 1 molecules such as virus-infected cells, cancerous cells, and cells originating from a tissue graft. The CD8 plus T cells require the help of mature CD4 plus T cells to proliferate and differentiate optimally. CD4 plus T cells browse the surfaces of professional antigen-presenting cells such as dendritic cells with their T cell receptors. Upon recognition of a cognate peptide MHC class 2 complex, they become activated and proliferate and differentiate into effector T cells. Effector T cells can be of diverse types. T helper type 1 Th1 cells are involved in the regulation of immune response to intracellular pathogens. T helper type 2 Th2 cells are involved in the regulation of immune response to several extracellular pathogens. Recent advances in immunology have identified two additional subsets of T helper cells, T helper type 17 Th17 cells, which derive their name from their ability to secrete IL-17 and play an important role in cell-mediated immunity and is possibly involved in defense against fungi. T follicular helper cells TFH, which play a critical role in humoral immunity and are involved in the maturation of B cells in germinal centers. The type of pathogen that is responsible for a given infection, intracellular or extracellular, virus, bacteria, protozoa, fungi, helminth, is the principal factor that determines which subtype of T helper cell will dominate the immune response. Each of these T helper cell subtypes releases a specific and different set of cytokines upon activation that in turn facilitates the activation of B cells. TC cell